Morning, everyone. Today we embark on a journey to demystify the intricacy of past year exam question for March 2023 for A-level students. Examining the past offers us a compass to navigate the challenges ahead, empowering us with insight and strategies to conquer the present. Let's dive into the trajectory truth of past exam, unlocking the key to success, and mastering the art of academic excellence. Question 1. The photomicrograph shows cells from a human blood smear where you have neutrophils and P and Q, which calculations show a correct method to calculate the actual diameter of the neutrophil shown in the photomicrograph in micrometers. The answer for this will be B. The actual length is actually length of line PQ in nm multiplied by 10,000 divided by magnification of photomicrograph. Number 2, a student calibrated an eyepiece reticule using a stage micrometer. Each division of a stage micrometer was 0.01 mm. With a multiply 10 magnification objective lens, 10 eyepiece reticule unit matches 10 division on the stage micrometer. The same microscope was used with a multiply 40 instead of A multiply 10 magnification objective lens to measure the diameter of an ovolus. The diameter of the ovolus was found to be 96 eyepiece reticule unit. The eyepiece lens was not changed. What is the best estimate for the diameter of the ovolus? To find the best estimate for the diameter of the ovolus, first you need to calculate the calibration factors for the eyepiece reticule. Then, with A times 10 magnification objective length, 10 eyepiece reticule unit match 10 division on the stage micrometer, which equals 0.nm. So the calibration factors is 0.nm divided by 10 eyepiece reticule unit. Then use the calibration factors to convert the measurement with the multiply 40 objective length. Since the eyepiece length was not changed, the calibration factors remain the same. The measured diameter of the ovolus is 96 eyepiece reticule unit, as given in the questions. So the actual diameter of the ovolus with the multiply 40 objective length is equal to 3.96 eyepiece reticule unit multiply 0.1 mm and divided by 10 eyepiece reticule unit, and you get the value of 0.96 mm. So the answer for number 2 is A. Which statements are correct for the chloroplast and also for mitochondria? We can see here, the first one is they contain ETS ribosome. Both is actually organelle, contain their own ribosome, which are 70S in size, but resemble ATS ribosome in sedimentation rate. This is occurs for both chloroplast and mitochondria. Number 2, they can transcribe their circular DNA, also happen in the both organelle. So chloroplast and mitochondria organelle have their own circular DNA, molecules, and machinery for transcription. Number 3, they can translate mRNA. Both is actually chloroplast and mitochondria. Organelle have ribosomes and translation machinery to synthesize protein from mRNA transcript. And number 4, they are enclosed by double membrane. This is a slightly different between chloroplast and mitochondria. Chloroplasts have more complex structure. They have an inner and outer membrane, And they also have an additional membrane system called thylakoid within their interior. Meanwhile, mitochondria are indeed enclosed by a number membrane. So the answer for this is 1, 2, 3, and 4. The electron micrograph shows some cell from a root, which cell structure is not usually found in a cell from a root. The answer is D. Dialysis with skin tubing is an artificial partially permeable membrane with pore sizes of approximately 2.5 nanometers. Glucose molecules have a diameter of about 1.5 nm and can pass through the pores in a membrane. What else can pass through the pores? First, bacteria, 2, hemoglobin, 3, ribosomes, and 4, fructose. Based on the given pore size of approximately 2.5 nanometer, glucose molecule can pass through the pores. However, items like bacteria and ribosomes are typically larger than 2.5 nanometer, so they couldn't pass through. Hemoglobin is a large protein molecule found in the red blood cells and is also too large to pass through. Fructose, being smaller than the pore size, could potentially pass through the membrane. So the correct option would be fructose, which is 4 only. The answer is D. What is present in all viruses? All viruses contain genetic material in the DNA or RNA, which could be made up of their either ribose or deoxyribose sugars. Adenine and thymine are specific nitrogenous bases found in DNA, while ribose and deoxyribose are sugar found in the backbone of nucleic acids. Since viruses can have either DNA or RNA, the correct answer is not specified. However, if we had to choose from the given option, ribose, deoxyribose, adenine, and thymine, the most correct option or the most correct answer is adenine, which is number 6 is C. To estimate the concentration of glucose is an unknown solution. Equal volume of a range of known concentration of glucose where each mixed with the same excess volume of dynamic solution. After mixing the solution, we place in a thermostatically controlled water bath at 19 Celsius for 3 minutes. The unknown solution was then treated in the same way and the color of the known and unknown solution compared. What is the independent variable in this procedure? The independent variable is the one that is intentionally changed or manipulated in an experiment. In this procedure, the concentration of glucose in the known solution is varied intentionally to observe its effect on the final color of the solution. Therefore, the independent variable in this procedure is concentration of glucose. The answer will be A. Which statement are correct for amylose and also for amylopectin? Both amylose and amylopectin are types of carbohydrate found in starch. So statement 1 is correct for both. They are carbohydrate molecules. Additionally, they are both formed by condensation reaction, where monosaccharides link together to form polysaccharide. So statement 2 is also correct for both. They are formed by condensation reaction. However, they differ in structure. Amylose is a linear molecule, so statement 3 is also correct for amylose, which is they are linear molecule. Amylopectin is branched, not linear, so statement 3 is incorrect for amylopectin. And both amylose and amylopectin contain alpha 1 and 4 glycosidic bond, so statement 4 is correct also for both. They contain alpha 1 and 4 glycosidic bond. And therefore, the correct statement for both amylose and amylopectin are they are carbohydrate molecules, they are formed by condensation reaction, and they contain alpha 1, 4 glycosidic bond, which is the answer is 1, 2, and 4, which is B. Which statement is the correct comparison between saturated triglyceride molecules and unsaturated triglyceride molecules of approximately the same molecule masses? The correct answer is unsaturated triglyceride have more double bond and fewer hydrogen atom than saturated triglyceride. In unsaturated triglyceride, some of the carbon-carbon bond are double bond, which means they have fewer hydrogen atom than saturated triglyceride, where all carbon-carbon bonds are single and saturated within hydrogen atom. The answer will be B. The diagram represents a molecule from a cell surface membrane, which a description of one of the label is correct. D is actually a phosphate group at the hydrophilic end of the molecule. Which row correctly show level of protein structure that can be held together by each type of interaction? The correct answer is C, which is hydrogen bond is the secondary and tertiary structure. Hydrophobic interaction, where is a tertiary structure, and primary and tertiary structure will be considered under covalent bond. Which molecule contains at least 3 double bonds? The answer will be D. Which diagram correctly shows hydrogen bonding between 2 water molecules? The answer will be B. Which statement describes an example of an extracellular enzyme? The option is amylase saliva is an enzyme that catalyzes the breakdown of starch in the mouth. Amylase saliva is an example of extracellular enzyme because its function outside of the cell, catalyzing the breakdown of starch in the mouth. Which row is correct for enzyme that catalyze reaction using the log and end hypothesis? The correct row for enzyme that catalyze reactions using the log and key hypothesis is B. The effect of the enzyme on the activation energy of the reaction being catalyzed. Lower the activation energy, shape of active site in comparison to the substrate, which is the answer, is a complementary before, during, and after the formation of an enzyme substrate complex. A scientist investigated the rate of breakdown of hydrogen peroxide. 4 experiments were carried out using different mixture, which is substrate only, substrate plus enzyme plus competitive inhibitor, substrate plus enzyme plus non-competitive inhibitor. The results are sketched in the graph. Which row show the correct line for 2 of the experiment mixture? Substrate only is referring to the 4, where you have substrate concentration. Okay, and then at the y-axis is the rate of breakdown of hydrogen peroxide. Substrate plus enzyme plus competitive inhibitor is referring to condition of 2. Which of these substances can pass directly through cells membrane without using a carrier protein or a channel protein? We have Ca3+, CO2, C6H12, and O6. The answer is CO2 can pass directly through the cell surface membrane without using a carrier protein or a channel protein. So the answer will be 2 only. When happened to the surface area to volume ratio of a cube, when the length of each side is double, when the length of each side of a cube is double, the surface area increases by a factor of 4, while the volume increases by a factor of 8, 2 to the power of 3. That's why the answer of 8. Therefore, the surface area to volume ratio decreases. So the correct answer is A. The ratio decreases by 4 times. Which event are part of the mitotic cells cycle? Interphase, telophase, or cytokinesis? The events that are part of the mitotic cell cycle include interphase, telophase, and cytokinesis. The answer will be 1, 2, and 3. The estimated total number of red blood cells in the human body is 2.5 times 10 to the power of 13. It is estimated that each day, 2.5 times 10 to the power of 11 red blood cells are removed from the circulation and are replaced by stem cells in the bone marrow. Which percentage of the total number of red blood cells is replaced each day? To find the percentage of red blood cells replaced each day, you can divide the number of red blood cells replaced each day by the total number of red blood cells in the body. Then multiply by 100 to get the percentage. So the calculation is 2.5 times 10 to the power of 11 divided by 2.5 times 10 to the power of 30, then multiply by 100 to get the percentage. So this simplifies to 10 to the power of 11 divided by 10 to the power of 30, then you multiply by 100, you get the value of 1%. Thus, approximately 1% of the total number of red blood cells are replaced each day. The answer will be C. DNA polymerase catalyzes condensation reaction between molecules during semi-conservative replication of DNA. What is joined by DNA polymerase? DNA polymerase catalyzes the formation of phosphodiester bond between nucleotides during DNA replication. So the correct answer is nucleotide and nucleotide. The answer will be C. Two polynucleotide strands make up a DNA molecule. Which description is correct? The correct description is B, where the percentage of cytosine is the same as that guanine in the whole molecule. This is known as Kaga rule, where the percentage of adenine equals the percentage of thymine, and the percentage of cytosine equals the percentage of guanine in the DNA molecule. 23. The PDX1 protein is found in a wide range of animal species, where it is regarded as essential for normal metabolism. In sand rats, the gene coding for the PDX1 protein has a much lower proportion of A and T nucleotides than PDX1 gene from other animals. Finding the complex DNA sequence of the PDX1 gene in sand rats has been difficult. Sequencing involves splitting of nucleotide one at a time from single-stranded DNA. What could account for a difficulty in finding the DNA sequence of the PDX1 gene in sand rats? The difficulty in finding the DNA sequence of the PDX1 gene in sand rat could be due to the strength of the hydrogen bonding between the two strands of the PDX gene is unusually high. This high strength of hydrogen bonding could make it challenging to separate the strand of the DNA during sequencing, which result in difficulty in determining the complete DNA sequence. And the answer will be one only. The PDX1 gene is present in only a small proportion of nuclei. 24. Different tissue in a plant were supplied with a radioactively labeled substance to identify which tissue were actively synthesizing mRNA. Which radioactively labeled substances would be suitable for this experiment, whether adenine, uracil, inorganic phosphate, or ribose. The suitable radioactively labeled substances for identifying actively synthesizing mRNA in plant tissues would be those involved in RNA synthesis. So among the options provided is adenine, is one of the nitrogenous bases found in RNA, so radioactively labeled adenine could be incorporated into newly synthesized mRNA. Number 2 is uracil. Uracil is another nitrogenous base found in RNA, specifically in place of thymine, so radioactively labeled uracil could also be used to identify newly synthesized mRNA. Therefore, the option 1 and 2 would be suitable for this experiment. So the answer will not be listed in either A, B, C, or D. 25th, which role correctly matches the structure and function of foliar sieve tube element? The answer for this is B. For the structure, is an end wall modified to form sieve plates, and for the function, is to slow down the rate of transport of solutes. Which features of xylem vessel element help adhesions during transpiration? So, a lignin form a complete secondary wall. The answer will be A. 27. Mass flow in the bulk movement of material from one place to another. How many of the vessels listed carry fluid by mass flow? We have artery, folium, sieve tube, vein, and xylem. Xylem vessel. The answer will be B. 2. Among the vessels listed, the folium, sieve tube, and xylem vessel carry fluid by mass flow. 28. Cellulose lignin and suberin are components of various plant cells wall. Description of the cell wall component are listed. 1. The component of a Casparian strip. Number 2. Redirect water into a simplest pathway. Number 3. A hydrophobic component of the cell wall. Number 4. Water interact with this molecule in the apoplast pathway. Which role correctly matches 3 of the description with the cell wall component? The correct answer or the correct matches would be C. For the cellulose, it's applicable for water interact with this molecule in the apoplast pathway. For the lignin, is actually a component of the Casparian strip. And for submarine, is actually a direct water into a simplest pathway. The arrow shows the direction of water movement across a plant root from water in the soil to the xylem. Which arrow show water movement only in the apoplast pathway? It should be 2 and 4. Which role correctly identify sources and sink of sugar? In plant, sources are where sugar are produced or released, while sink are where sugar are consumed or stored. Root cells absorbing mineral ions are typically sink because they consume resources mineral ion from the environment. And the storage cell of a seed that is starting to grow act as a source. They provide energy for the growing seedling. So the correct answer would be C. Root cell absorbing mineral ion is a source, and storage cell of seed that is starting to grow is also a source. The photomicrograph shows a section through a tissue with an artery. Which role correctly show the types of artery and whether the blood inside the artery is oxygenated or deoxygenated? Type of artery is a muscular and the blood is deoxygenated. Heart surgery may cause a decrease in the transmission of impulses in a Purkinje tissue to the right side of the heart. What is a possible effect of this decrease? The possible effect of the decreased transmission of impulses in the Purkinje tissue to the right side of the heart would likely result in the muscle of the right ventricle would contract slightly more slowly than the muscle of the left ventricle. And this is actually because of the Purkinje fibers are responsible for rapidly transmitting electrical impulses to the ventricle, coordinating their contraction. If there is a decrease in transmission, it would affect the right ventricle contraction speed more than the left ventricle. The answer for this would be B. 33. The diagram shows a network of blood vessels that supply blood to a muscle tissue in the human body. And what is a correct comparison between the X to the blood at X and the blood XY when the muscle tissue is at rest? The answer will be blood pressure at X compared with the blood pressure at Y is a higher. And water potential of blood at X compared with water potential of blood at Y is also higher. Scientists have shown that the oxygen dissociation curve for hemoglobin of smaller mammals are to the right of those of larger mammals. What does this suggest about the hemoglobin of smaller mammals? The fact that the oxygen dissociation curve for hemoglobin of smaller mammals are to the right of those of larger mammals suggests that it saturates with oxygen at the lower partial pressures of oxygen than the hemoglobin of larger mammals, which means that The smaller mammal hemoglobin reaches maximum saturation with oxygen at lower oxygen level as compared to the larger mammal, which is indicate higher affinity for oxygen binding. So the answer will be B. 35. What is an effect of an increased concentration of carbon dioxide in the blood? An effect of an increased concentration of carbon dioxide in the blood is A, increased movement of chloride ions out of the red blood cell. This is known as the chloride shift, which help to maintain electrochemical equilibrium across the red blood cells membrane in response to increased carbon dioxide level. The answer would be the increased movement of chloride ions out of the red blood cells. The answer would be A. Two of the requirements of an efficient gas exchange system are a large surface area and a short diffusion distance, which role correctly describe how everly are adapted to meet this requirement. So for the large surface area, alveoli are folded and interconnected, and for the short diffusion distance, walls of alveoli are only one cell thick. What define infection diseases? The definition that the best define infection diseases is symptoms are caused by a pathogen that is transmitted from one host to another. Infection diseases are caused by pathogenic microorganisms such as bacteria, virus, fungi, or parasite that can be transmitted from one individual to another. 38. A hurricane destroyed a large town of an island. People move away from the town and set up tents where sanitation is poor. Which disease is more likely to spread within a week of a change in living condition? The sudden change in living condition, the disease most likely to spread within a week for this scenario is cholera. Cholera is a waterborne disease commonly associated with the area with poor sanitation and contaminated water sources, and the crowded living condition in tent without proper sanitation facilities create an environment conducive to the rapid spread of cholera. The answer would be A. Which statement correctly explains why viruses are unaffected by penicillin? The statement that correctly explains why viruses are unaffected by penicillin is D. Penicillin only blocks peptidoglycan synthesis. Penicillin interferes with the synthesis of bacterial cell wall by inhibiting the formation of peptidoglycan, a component specific to bacterial cell wall. And viruses do not have the cell wall made of peptidoglycan, so penicillin does not affect them. The answer will be penicillin only blocks peptidoglycan synthesis. Number 40, which processes characterize the mode of action of phagocytes? We have antibody production, receptor binding, 3 endocytosis, 4 exosynthesis, and hydrolysis. The process that characterize the mode of action of phagocyte include receptor binding. Phagocyte recognize foreign particles through receptor on their surfaces, and endosynthesis. Phagocyte engulf the foreign particles through the process called endosynthesis. And 5, we have hydrolysis. Phagocyte break down engulf particles using enzyme through hydrolysis, which is the answer will be 2, 3, and 5, where the correct answer does not exist in any of these answers, whether A, B, C, or D. With that being said, that's all for today's presentation. And I would like to thank you for your preparations and support all this while. And please subscribe to Jerome Study Lab YouTube channel. Thank you very much.